Welcome to Genetics 9 Inbreeding. And what we're talking about mating systems. Previously in this chapter we discussed how we select animals to be parents for the next generation. Now we're going to try to figure out how do we match up the different males with the different females. In one mating system would be inbreeding, either intentional or accidental. And how do we calculate the amount of inbreeding? So we're going to cover the following types of mating systems in this section. Basically inbreeding, intense inbreeding, and line breeding is also inbreeding, but the purpose is a little bit different. With line breeding, generally we want to concentrate the genes of an outstanding individual while still keep inbreeding relatively low. Inbreeding, what's the definition? Inbreeding, if the parents are related, the offspring are going to be inbred. And how do you tell if they're related? They have to have common ancestors back in their pedigree. And how far back do we trace? So here are the two parents. Do they have a common ancestor? So in this case, the sire and the dam are related. And here's the sire and dam. I like to circle those because it's how the sire and dam are related, which makes the offspring inbred or not. So here we have the common ancestor A. In fact, we made it a son of A times a granddaughter of A, and that's going to make the offspring inbred. Now, in this example, we're just have gone back just a couple of generations. Here the parents again, and we went back a couple of generations. We found this common ancestor A. Obviously, if we go back more generations, we might find additional common ancestor back there. As you go back each additional generation, you're doubling the number of ancestors. So a lot of times we have to decide how far back we want to go in the pedigrees to determine relationships of those parents. So we may decide we're going to go back 4 or 5 generations and look for common ancestors. To calculate the inbreeding for dairy cattle genetic evaluations in the U.S., they go back to an ancestral base year of 1960. So each line might be a little different. In some cases, they're going back 30 generations to go back to where this last animal here was born in 1960. So that's another way to do it. And a lot of the national genetic evaluations, they just tend to go back to a certain year and stop. The further you go back in the pedigree, the smaller the contribution to inbreeding any particular ancestor makes. So really, in a lot of cases, there's no reason to go back too many generations. Alright, so you've determined that an animal is inbred because of a common ancestor, but how inbred is the animal? The animal can be highly inbred or not very inbred at all. So the degree of inbreeding we measure with the inbreeding coefficient. A capital F sub X is kind of common nomenclature. And it actually, the number does stand for something. It's the average decrease in genetic heterozygosity due to relationship of the parents. So at any one loci, you can have a heterozygous genotype or you can have homozygous genotypes. If an animal is shown to be 10% inbred, it means that the number of heterozygotic genotypes have been decreased by 10%. The inbreeding coefficients, the average decrease in genetic heterozygosity, you're going to have more homozygous genotypes. Well, there are several different ways to measure or calculate that inbreeding coefficient number. We're going to discuss two different ways here. There are other ways used in the industry as well. These are two of maybe the simpler ways. And first one here is from the known relationship of the parents. And again, it's how the parents are related that's going to determine the inbreeding coefficient of the offspring. So if we made an individual times a parent, that's going to be pretty inbred, 25%. If full sibs are mated, inbreeding coefficient of the offspring would also be 25%. If you made half sibs, what's a half sib? It's two individuals that share the same father or same mother, but not both. Inbreeding coefficient's 12.5%. Individual times a grandparent, and certainly with artificial insemination where we can freeze semen, that's certainly possible to do, but inbreeding is going to be 12.5%. We do a son of a sire times a granddaughter of sire, 6.25%. Well, how much inbreeding is too high and when isn't it too high? There's no magic number, but usually we like to, it's been kind of common practice, keep the inbreeding less than 6%. That's kind of common. Or another way to say it, if it's illegal in humans, then maybe we shouldn't do it in animals either. Example of a son of a sire times granddaughter of a sire. So again, here are the two parents. Let's just circle those. We've got the son of this sire times the granddaughter of the sire. So here's A here. Here's its son. And here's the granddaughter. And this individual here is going to be inbred by 6.25%. Probably higher than we would a lot of times like, but not terrible. And again, it's how the parents are related that's going to make X inbred. What is the inbreeding coefficient in this pedigree? It appears the two parents are related. And if we go back to our chart, it seems we've got a grandson times a granddaughter, and that inbreeding coefficient is 3.12%. And 3.12% is not terrible. If we can choose a different sire for this particular dam that's not related at all, we might be better off, but you would not expect very detrimental characteristics coming from this low of inbreeding. So here's another way to calculate inbreeding from bracket pedigrees. Works on the principle that the amount of inbreeding due to common non-inbred ancestor is dependent on total number of generations. The common ancestor is back from the parents. So again, we're counting back from the parents. How many generations are we back? One generation, it's going to be 25%. Two generations, 12.5. And as we go down here, you can see every time you go another generation back, you're dividing the amount of inbreeding by 2. So if you go back 6 generations, 0.78, essentially with that low inbreeding coefficient, most people would say that that animal is not inbred at all. So here we have a simple little pedigree, I guess, but how inbred are we in this particular case? So here are the two parents. We want to know the inbreeding coefficient of X here. So first we need to draw a circle around the parents. Always a good idea. So we'll locate the common ancestor, and I like to circle the common ancestor. And we may have more than one, but we've got one in this case, and then we can count the generations back from the parents. So here's that common ancestor again. From the parent to the common ancestor on top, it's the same individual. So number of generations there is 0. On the bottom from the dam, it's the sire. We're back 1 generation total. So again, you count from the parents. Students will make this mistake. They'll start counting from the offspring. But then you're going to be wrong. It's how the parents are related. We count back from the parents. So then we go back to our chart here. We are 1 generation back. So our inbreeding coefficient is going to be 25%. So here's a pedigree that we've seen already. Son of a sire times a granddaughter, and we've calculated before from a chart that it was 6.25%. Well, let's count the generations back and see if we also come up with that 6.25%. So here we are. Here are the parents again. Here's our common ancestor. You can see on top here we're back 1 generation. On the bottom we're back 2 generations, 3 generations total. We're back 3 generations total, so we can look on our chart here. 3 generations, inbreeding coefficient 6.25%. Well, pedigrees can get pretty complicated. For this particular course, this is about as complicated as we're going to get. We've got the 2 parents here. A is a common ancestor. And then we've got another common ancestor totally unrelated to A, and that's B back here. So basically we've got 2 sources of common ancestor that make the 2 parents related, and we've got to take each into account kind of separately. So we've got inbreeding due to A and due to B. So first due to A, back for 1 generation on top, 1 on the bottom, total of 2. Look at our chart, that'd be 12.5%. Amount of inbreeding due to B is going to be quite a bit less. So we've got 2 generations on top, 2 on the bottom, total of 4. Look on the chart, 3.12. So we've got an individual here that's inbred by 15.62%, and that would be considered pretty high inbreeding. There's a lot of different ways to draw a pedigree, so here's another example. We want to know the inbreeding coefficient of this individual. Circle the parents. How are they related? I see 1 ancestor here, back here 2 times. Back 1 generation here, 1, 2 here, total of 3 generations. But we draw a line right through the look at the pedigree of the father, look at the pedigree of the mother, we see that they do not share any common ancestors. So in this case, the S and D, the two parents, are not related. Since they're not related, the inbreeding coefficient of X is going to be 0. X is not inbred. Now the sire is inbred. The sire is inbred by quite a bit, but you do not inherit inbreeding from one generation to the next. It's how the parents are related that's going to cause the individual to be inbred. So we can see S here is inbred. Circle S is two parents. Where we got a sire times a daughter, individual times a daughter, that inbreeding coefficient is 25%. So the father here is very inbred, but the offspring is not inbred at all because the father is not related to the dam. Well, here's another little example here for us to calculate the inbreeding coefficient. Again, here are the two parents. I see a common ancestor back here, only one that's denoted in the pedigree. Let's see, on top here we're back 3 generations. 3 generations at the bottom, total of 6 generations back. I think the inbreeding is going to be relatively low. Yeah, so we're 6 generations back. We look at our chart and its inbreeding coefficient is less than 1%. It's only 0.78%. So this little chick here is inbred, but not really. You know, you're less than 1%, the inbreeding's pretty low. So again, that's why we calculate inbreeding coefficient, so we can quantify it. You might say this chick is inbred, just like one that is 25% inbred is inbred, but this inbreeding coefficient is extremely low. And I can bring that up back many years ago. There was a Kentucky Derby winner that went on to race successfully in other races until the horse broke down, broke its leg, and had to be destroyed. And of course, that's not good when a horse breaks its leg, and so it made headlines, and several people blamed the fact that the horse broke down because it was inbred. And thoroughbred pedigrees are readily available, so I checked the pedigree and found out that yes, that horse was inbred, but the inbreeding coefficient on that horse was 0.39%. You know, half of what we got here, a third of 1%. Now, a third of 1% is very, very, very little inbreeding, and you wouldn't expect that low level of inbreeding to cause any problems at all. So yes, the horse was inbred, but at extremely low level, and that's why we calculate inbreeding coefficients. How inbred is that animal? So here's another pedigree that we have here, a little bit of a trick pedigree. See what you can do with it. Might hit the pause button and work things out before we give you the answer. So look at it carefully and see what you come up with here. So here we have the parents, common ancestor B here. So B is back 2 generations on the top, B is back 2 generations on the bottom, total of 4 generations. And that inbreeding is 3.12%, I believe, if we look at the chart. Now we also got Z back here as a father of B. But as far as Z back there goes, we've already accounted for Z when we accounted for B here. So Z, after accounting for B, is not going to contribute to the relationship of the two parents in this case. So we accounted for B. B has to have the same father, top and bottom on the pedigree. B has to have the same mother. In fact, as far as grandfather back here, the father of Z has to be the same in both cases, so that individual is going to be the same. The grandmother is going to be the same. And if we extend out B's pedigree, it has to look exactly the same top and bottom, going back 10 or 20 or more generations. It has to be the same. Now, as far as relationship, we've accounted for it when we did B. If we count all these other ancestors, well, we're going to be counting forever, and that inbreeding is going to get up to over 100%, to which, no, we don't do that. Once we accounted for B, we've accounted for B's full pedigree here. So don't overcount. You stop at this individual right here. Anyway, inbreeding 3.12%, not all that high for this particular chicken here. Generally, we try to avoid inbreeding, and inbreeding is generally detrimental, not equally detrimental for all traits. It tends to be most detrimental for reproduction, health, and vigor. And we call this inbreeding depression. Inbreeding depression does not affect all traits equally. Inbreeding has really no effect on mature size. Inbreeding usually has no effect at all on show-ring type. In fact, it may enhance show-ring type sometimes. So basically we're saying that you cannot look at the animal and say, well, that animal looks like it's inbred. You really can't do that because some of the best looking animals may be quite a bit inbred. But inbred animals do tend to have a few more problems as far as fertility and overall disease resistance. Now I threw this particular picture in here, and just because it's kind of cute, I guess, but I do not know if this dog is inbred or not. It very well could be. A lot of dogs do have some inbreeding when you go back in the pedigree, but no, this dog here could be quite outbred. I don't know if it's inbred or not. But effects of inbreeding we can uncover undesirable recessive genes. If this dog would have a recessive gene here, this little N, we can't tell by looking at the dog, but if we breed this dog to a relative, we might get those two little N's in an offspring, and then we know we have that undesirable recessive there, and maybe modify our breeding programs to minimize this effect. So we've got this mating here, and obviously X is inbred here by 25%, did a daughter times a sire. Now just by chance this sire is a carrier for this undesirable recessive gene here. Now this sire is completely normal in appearance because this gene is recessive. As long as you got one normal gene, the animal is normal. So we may not even know that this genetic defect even exists in our population. But if we do some inbreeding, it may come to the forefront. So this N here can be passed on to this individual, And this individual coming down that end is, there's a 50% chance. Let's do 0.5. You got a 50% chance that the N is going to be passed on, little n, recessive genetic defect gene is going to be passed on from the parent to the offspring. How about from the dam? What's the chance of getting another little n here so that we have an affected individual and we find out that we do have this recessive back here? Well, from this generation to this generation, it's 0.5, and from here to here, it's another 0.5. So this second end there is going to come with a probability of 25%. So the probability of uncovering this recessive condition with this one inbred offspring then is going to be 0.5 times 0.25 or 0.125. So we've got a 12.5% chance of discovering that this ancestor back here is this carrier for this undesirable recessive. Well, another effect of inbreeding is that we may get heterosis, which is a good thing. We'll talk about heterosis in the crossbreeding section. But heterosis is generally an advantage that you get when you cross different breeds or cross different non-related inbred lines. So the key thing here, if you're going to cross inbred lines, those inbred lines need to be not related to each other. So inbred lines have been developed in corn and poultry going back almost 100 years. In corn and poultry, it's maybe a lot easier to develop inbred lines than in our larger species. Another effect of inbreeding, it increases homozygosity or homozygous genotypes. So in our example here, we've got 3 possible genotypes. We've got the big A and the little a in our population. And well, let's go through an example here and see what would happen with high levels of inbreeding. So we're going to assume we got frequency of the big A at 0.9, frequency of the little a here at 0.1. If you put that in Hardy-Weinberg equilibrium, P is 0.9, so frequency of the homozygous dominant genotype, 0.81, 2PQ is 0.18, and Q squared or 0.1 squared is 0.01. Percent that are homozygous is 0.81 plus the 0.01, 81%. And I really should have another column here and percent heterozygous genotypes. That would kind of be a good thing to look at too. It would just be 18% then. Percent heterozygous genotypes is the big A, little a. That's the heterozygous one. We're going to assume we got no inbreeding starting off here. To show the effects of inbreeding, we're going to use corn plants. And the reason for corn plants is that we can inbreed a lot faster. So we've got this corn plant here. We can take the pollen, drop it down on the silk, and so we just bred the corn plant to itself here. So these kernels of corn, the germplasm within these kernels is going to be inbred by 50% after one generation here. So we can do it pretty fast and see what happens. So the homozygous corn plants, of course, they can only have homozygous offspring. They're only transmitting the big A. And the same for the homozygous recessive. all those offspring are going to be 0.01. So, and we're saying this is the percent of genotypes out there, so these 81% produce themselves. This 1% produce themselves. Now the heterozygous individuals don't produce themselves, because if we put this into a Punnett square, we find that a quarter of the offspring are going to be two big A's, a quarter are going to be two little A's, and one half are going to be heterozygous like the parent. So here we have it. So this individual is not going to reproduce identical to itself. Only a half are going to be heterozygous like the parent. 1/4 homozygous dominant, 1/4 homozygous recessive. So we have changed the genotypic frequencies from our one generation of inbreeding. So second generation, then we just added this together. Amount of heterozygous genotypes is 1/2 now what it was. We've got more of the homozygous genotypes. Frequency of the recessive allele has stayed the same. So a lot more in the homozygous recessive genotype, but we've lost that recessive gene from the carriers. So that balances out. Percent homozygosity now is 91%. Percent heterozygosity went from 18% down to 9%. Inbreeding coefficient after one generation of selfing is 50%. We've lost 50% of the heterozygosity. Well, let's do it another generation. So we've gone a second generation of selfing now with these corn plants. Notice the heterozygosity now, we've lost 75% of the heterozygosity, and that's kind of definition of inbreeding for coefficient, percent heterozygosity lost. A lot more homozygous genotypes, especially for the homozygous recessive. We went from 1% being homozygous recessive to 7.7 or almost 8%. So inbreeding can really increase this homozygous recessive genotype, especially if you're starting at a low frequency. It can go to almost nothing to quite high where it's going to be noticeable. Another thing we might notice here is that what's happened to the frequency of that recessive allele, and that has stayed the same. Homozygous recessive genotype has really increased, but the frequency of that recessive allele stayed the same. It just has come a lot fewer carriers in our population. There are more in the homozygous recessive genotype. So just want to emphasize this again. Inbreeding does not change gene frequency. The frequency of our little a started out at 0.1, it remained at 0.1. Frequency of the dominant big A started out at 0.9, it's still 0.9. Sometimes in the popular press, when a new recessive genetic defect shows up, you might say, well, the frequency of this recessive allele increased because of inbreeding. No, it didn't increase because of inbreeding. It may have increased because you selected an individual to use as a parent that contains the recessive gene, but inbreeding by itself does not change gene frequency. It's selection that changes gene frequency. Several times I've read that inbreeding cause this particular mutation when a new mutation is discovered. And no, inbreeding does not cause mutations. Radiation and mistakes in DNA replication causes mutations, but inbreeding by itself does not cause mutations. You know, what is a mutation? Well, a mutation is a change in the genetic code or change in DNA. Mating two related animals is not going to cause a change, a physical change in that DNA. Well, this is just an example of an inbred cat, just in case you haven't seen one before. But especially farm cats, you probably get quite a few that are inbred because the tomcats are around and they mate with each other. So it wouldn't be unusual, I don't think, for colonies of cats to become fairly inbred. What is line breeding? Line breeding is inbreeding, but it has a specific purpose to it. When you're line breeding, we're trying to concentrate the genes of an outstanding individual. As you go, we got an ancestor back there that's pretty great. Well, each generation, that particular ancestor's genetics are going to be diluted by 1/2 if we don't inbreed. So we're going to inbreed to that common ancestor because we don't want to lose those genetics after several generations. Generally, we don't want high inbreeding. A lot of times with inbreeding, successful programs with line breeding, they generally want to keep the inbreeding coefficient to maybe 10% or less. How much is line breeding practiced today in our food producing animals? It's really not practiced. It was practiced some in the past if you go back 60, 70 years, but now with the calculations of estimated breeding values and genomics, we're probably better off not mating related animals. It's still practiced some in the for the show industry. Inbreeding is not detrimental, generally the showing type, and so there are a lot of outstanding animals in the show industry, especially maybe in the dog industry, that tends to be maybe fairly inbred. So here we have a pedigree of a line-bred individual. So when you first glance at this pedigree, you might say this individual X is extremely inbred because Z occurs 4 times back in the pedigree. And I'm not going to illustrate how we calculated the inbreeding coefficient, but trust me, the inbreeding coefficient of X here is 7%. And like I say, with line breeding, we generally like to keep the inbreeding at 10% or less. So anyway, Z is back here several times. If you know, A got half of Z's genetics, M got 1/4 of Z's genetics, but we're back here several times. So if we look at X and count the relationship back to Z, what percent of Z's genes do we have in X? We find that X has 3/8 of Z's genes. X is line bred to Z. Now, for a successful line breeding program, as we're concentrating the genes of Z, we want to do selection from generation to generation as well. So here we've got 4 offspring of Z. And hopefully as we went along there, we got a lot more offspring, and we selected the best offspring of Z for the next generation. In each generation, we select the best offspring to produce X here. So line breeding, if you're going to do it successfully, it has to be in conjunction with very good selection along the way too, to really get an outstanding individual here that's going to have the best of these genetics. And I guess this is the end of this video for inbreeding and line breeding.